Okay, let's dive into this. Have you ever actually looked at a tiny medicinal pill and wondered, how do we really know what's inside it? I mean, really, truly know. In this explainer, we're going to look at pharmaceutical analysis not just as dry chemistry, but as a high-stakes forensic investigation. Imagine the analytical chemist as a master detective, hunting down the absolute truth hidden right there at the molecular level, ensuring that the medicine you take is exactly what it claims to be.
To figure that out, here's our roadmap for today's investigation. First, a quick intro to pharmaceutical analysis. Then, the art of titration. Third, advanced precipitation methods. Fourth, gravimetric analysis. And finally, number 5, defeating analytical errors.
Right, starting with Section 1, Intro to Pharmaceutical Analysis. Let's just briefly set the stage for why this entire field even exists and why getting this right is quite literally a matter of life and death. On paper, pharmaceutical analysis is simply defined as the branch of analytical chemistry dealing with the study of pharmaceutical products. But in practice, this is the rigorous, totally uncompromising scientific process of verifying a medicine. We have to prove, beyond a shadow of a doubt, that a drug is safe, that it's going to be effective, and most importantly, that it's completely free of impurities or false leads that could harm a patient. It is the ultimate quality control check.
Moving right along to Section 2, the art of titration, standard interrogation. If we're the detectives, well, this is our standard interrogation room.
Now, in any good interrogation, you need someone asking the tough questions. That's our titrant. The titrant is a solution of a known concentration, always sitting up top in the burette. You know, that long, graduated glass tube. You can really think of the titrant as our interrogator. It already holds the facts, it knows its own strength, and it drops into the scene, literally drop by drop, to extract the truth.
And on the other side of the table, we've got the titrant, which is also known as the analyte. This is our primary suspect. It's the solution of unknown concentration, and it always sits down below in the conical flask. It is just waiting down there to react with our interrogator, eventually revealing its true identity and exact concentration the second the reaction reaches its completion point. This visual brilliantly illustrates one of the classic setups, acidimetry. Check out this apparatus. We have a standard acidic solution up in the burette, our interrogator, dropping down into a basic or alkaline substance waiting in the flask below. We use the known acid to determine the exact unknown concentration of the base. But what if our suspect is an acid? Easy. We just flip the script with alkalimetry. Notice how the entire setup is reversed here? Now our interrogator up in the tall burette is a standard base, and it's dripping down into a flask containing an unknown acidic analyte. What's really fascinating about this inverse relationship is honestly how simple it is. Acidimetry uses a standard acid to interrogate a base, while alkalimetry uses a standard base to interrogate an acid. The tool you choose entirely depends on the nature of the suspect you're investigating. You always fight fire with water, so to speak, using opposites to find the balance, or what chemists call the equivalence point. So, everything we've talked about so far relies on water as the environment for our interrogation. But what if our suspect simply refuses to cooperate? Like, what if the pharmaceutical compound is completely insoluble in water? Or worse, reacts violently with it? That is exactly when we pull out our specialized backup plan, non-aqueous titration, when standard water-based methods fail, which often happens because the drug sample is just too weak of an acid or base to give us a clear, sharp reading. We completely change the environment. We dissolve the analyte in an organic solvent that doesn't contain a single drop of water. Sure, it's a bit more expensive, and we definitely have to be careful with volatile solvents, but it forces those stubborn organic compounds to finally reveal a sharp, highly accurate endpoint. Which brings us to Section 3, Advanced Precipitation Methods, forcing suspects out. Because sometimes our suspects are just so elusive in liquid form that we literally have to force them out of hiding by turning them into a visible, solid mass. A major technique for this is argentometric titration. The name sounds intense, but it simply comes from argentum, which is just the Greek word for silver, hence why silver is Ag on the periodic table. In this method, our chemical weapon of choice is silver nitrate. When silver hits certain unknown halides, like chlorides or bromides, it reacts incredibly fast to form an insoluble solid precipitate. Let's see how this builds into an actual strategy with the famous Volhard method. Think of this as a brilliant two-step trap set by the analyst. In step 1, we don't just add a little silver,
We absolutely flood the scene with a known excess amount of silver nitrate to ensure every last bit of our suspect is captured into a solid precipitate. Then, in step 2, we perform what's called a back titration. We take potassium thiocyanate and use it to measure whatever excess silver is left over. By subtracting the leftover silver from the total amount we originally flooded the scene with, we can perfectly calculate exactly how much of our suspect was in that flask. It's an incredibly clever, indirect workaround.
Okay, moving on to Section 4, Gravimetric Analysis, Weighing the Evidence. Up until now, we've relied on volumes of liquids to find the truth, but sometimes you just need something more definitive.
Gravimetric analysis is basically the absolute method. Gravimetric literally just refers to mass. Here, we aren't looking at liquid color changes or titration endpoints. Instead, we completely convert our pharmaceutical analyte into a pure solid precipitate, filter it, dry it out, and physically weigh it on a highly precise scale. It is incredibly accurate and practically immune to things like temperature changes. But, as you can probably guess, it's painstaking. It requires perfectly clean glassware and a massive amount of time.
And it comes with a major risk: co-precipitation. This is essentially the ultimate false lead in our investigation. Co-precipitation happens when innocent bystanders, just random impurities in the solution, get dragged into the solid mass right alongside our main suspect during the precipitation process. If you weigh that contaminated solid, your entire mass calculation will be artificially heavy, and boom, your investigation is compromised.
So, to avoid the messiness of those unwanted solids, scientists use complex symmetric titration for certain targets, specifically metal ions. Instead of forming a solid, we use a complexing agent, the most famous one being a multidentate ligand called EDTA. Think of EDTA as this microscopic chemical claw. It reaches into the solution and perfectly wraps around the metal ions, trapping them in a stable, soluble complex. It's elegant, it's clean, and it gives a distinct color change without any risk of co-precipitation.
Finally, let's talk about Section 5, Defeating Analytical Errors, Analytical Flaws. Look, even the greatest detectives make mistakes, and a good scientist absolutely must confront their own blind spots. In any analytical operation, flaws are bound to happen.
So the crucial point here is how we actually define a mistake. In pharmaceutical chemistry, an error is simply the numerical difference between the true standard value of the drug and the observed value we actually got in our experiment. It isn't inherently a failure, right? It's just a measurable gap that we have to identify and close.
To tackle this, we divide errors into two main camps: systematic and random. Systematic errors are determinate, meaning we can track down their cause, like a faulty instrument or a bad procedure, and we can avoid them with careful planning. On the flip side, we have random errors. These are the indeterminate ghosts in the machine. They are completely uncontrollable, and their specific cause is totally unknown.
When we look closely at systematic errors, we find the core of today's focus, the 5 major flaws behind analytical operations. Number 1, personal errors. These are mistakes born of human inability, like an analyst who's slightly colorblind missing a subtle color change indicator. Number 2, instrumental errors, using defective or uncalibrated glass burettes. Number 3, reagent errors, relying on chemical solutions that secretly contain impurities. Number 4, additive errors, flaws that mysteriously remain perfectly constant regardless of how much sample you use. And number 5, method errors, simply selecting the wrong chemical procedure right from the jump. These 5 flaws are the ultimate enemies of precision.
To defeat them, we rely on a strict detective's checklist. We calibrate all apparatus beforehand. We might run an entirely independent method of analysis on the side just to double-check our work. We run a blank determination, meaning doing the whole experiment without the drug suspect even present, just to see if our reagents are hiding any chemical impurities. And we run a control determination, using a perfectly known standard to verify our setup is completely sound.
By strictly following that checklist, we can virtually eliminate systematic flaws. But remember those indeterminate random errors? The ones with totally unknown causes? They literally cannot be eliminated, not even by a perfect analyst, using the highest quality reagents and the best apparatus in the entire world. There will always be a microscopic margin of the unknown. So, knowing that absolute 100% perfection is scientifically impossible due to random error, how confident are you in the exact molecular dose of your next prescribed pill? It is a fascinating balance of rigorous science and acceptable tolerances. Thank you for joining me on this explainer, and keep questioning what's under the microscope.